Okay, well, I'm excited to be here today, and I know you are all tired, so just bear with me. I promise I have good animation for you. Today, I want to talk and really build on a lot of what has been discussed throughout the day and try to synthesize some of these ideas into take-home concepts, and also to add onto David's comments some other thought-provoking topics that I think we need to be thinking of if we really want to translate what we're doing in this multi-omic space into the clinic. So imagine the year is 2050 and you go to your EMR and you click on a little button and you get your own personalized health profile, complete with disease risk, your personal drug response profile, your overall 5-year trends, your biological age, your gut health, personalized nutrition, supplements, and exercise. This is what we have been waiting for, right? And so I think it's often important to look at the target when we want to see where we're going. And that's why I'm putting this up here, because this is where we're headed and how do we get there? And I think there's a lot of things we're doing right, but I also think there are things we need to consider as we move forward to get to this endpoint. So in thinking about various topics today, we can think about each one of the omics and different ways they can have contributed. And here's an example. Some people said polygenic risk scores work well, others said they didn't. But just think of, if we have all of our genome-wide association data online, we could quickly just run these algorithms and get our prediction risk for all sorts of diseases. Things like this could feasibly be pretty feasible to translate into the clinic relatively easily with just algorithms if we can develop robust ones. We also think about things like biological aging, and we've seen today that this is really a huge emphasis of all of chronic disease, is this concept of biological age and slowing acceleration of age. But I want us to stop and think about what a lot of the messages have been. And fundamentally, I just want us to look at this deconstructed car and hear the message of today, which has been, when we look at multi-omics, simply put, the sum of the parts are greater than the whole. And this has been the promise of today, right? That while we look at all these parts separately, if we actually put them together, that working car is much greater than the sum of the parts. And we've talked about this, and we've talked about this in this multi-omic integration framework, where we take the genome, and then we take the epigenome, and the transcriptome, and the proteome, and the metabolome. We talked about a lot. The microbiome, the exposome, and finally the phenome. And a lot of what we've discussed is looking at all of these together in some type of integrated framework, where we can then use multi-omic integration with systems biology, machine learning of some sort, to ultimately develop some personalized treatment. And that last little animation didn't work out quite right, but as you can see, just pretend those are all the personalized treatments. And this is a great concept. But I want us to stop and think. Now stare really hard, don't blink, and tell me the answer. The simple fact is, as we generate more and more omic data and we put it together, the picture becomes more and more complex. And the fact of the matter is, when all of that data are integrated together, very often we cannot see the forest through the trees. When we look at this integrated framework, and as you know, there was a time period when everyone was publishing these figures, there's really not much we can synthesize when it comes to disease. And fundamentally for clinical translation, while we use multi-omic integration to inform things, it needs to be synthesized down to something simple. It cannot be this complex. This is not going to work in the clinic. And while we talk about systems biology and machine learning, ultimately, how can we translate this into something that's actually usable in the clinic? And I want us to think about this. And one of the main things I want to say is that the elephant in the room is that machine learning and methodological approaches for this are really, really, really difficult. And machine learning is a catchphrase, but the methodology behind it is complex and not clear. We also need to be careful about what we're inferring using the various study designs we have. And while we want to look at these interrelationships and potential ways one thing modifies something else, it can be very complex when we're doing it all together. And what I want to talk about is ways that we can take that complex hairball and synthesize it down into something that is more tangible, more digestible, and simply more clinically translatable. And this is a figure of a recent analysis our group has done, just looking at microRNA and metabolomics in a WAS type analysis. The first picture was a black ball, but as we synthesized this down, and looked at things in terms of number of associations, looked at pathways, we could very easily see things sort of as dysregulation in sort of endogenous pathways, and importantly also important contributions from exogenous metabolites that could be modified. So immediately sort of putting into a framework, in this case for asthma, of how we may be able to use more complicated multi-omics in a clinical translation. So thinking about this, I also want us to think about several other things. Well, I've just demonstrated an example of how we can take that complex hairball and using methods distill it down to something that is more tangible and understandable. The other very important thing is we need to be able to reproduce this over and over again. We can't just do it once, we need to show the robustness of this. So thinking through how we do this, going from something complex to something more simplified and showing it's reproducible, I think it's important to go back to this picture and think about how we can get it. So we take, let's say each one of those different types of parts is a different omic, and we want to put this back together in the framework of a car that works, that may need to be tested a couple times. We might have a Ford, we might have a Porsche, we might have a Benz, I don't know. But how can we do it so we're able to do this in frameworks where things are a little bit different, with maybe different cohorts, different characteristics? And I will say to you today that I think one of the fundamental ways that we need to do this is not only through replication. I think that is essential, but when thinking of the sum of the parts and emphasizing this idea of multi-omic integration, I also want to emphasize the name of this conference, which is PAN cohorts, and think about the context of even essentially looking at cohorts as another way in which we can take that hairball and make it more robust by various study designs synthesized together to understand disease from etiology, development, characterization, all the way to the clinical translation. Now, I'm going to do this very quickly with going through just a few examples of just study design, starting from the beginning, starting from conception, but then also giving an example of where we go through that really tangible clinical translation and thinking of it like a doctor, not a PhD student or a PhD researcher. So let's just start at the beginning. I think one of the important things to remember is when we think of these different study designs, they tell us different things. And when we go all the way back to birth cohort studies, the fundamental principle there are these, prenatal environmental exposures that may have long-term ramifications on disease. And so this is fundamentally about disease etiology and potential modification very, very early in life. And just to highlight this, I'm not going to go through any of the actual publication, but in this particular study we did with one of our birth cohorts, looking at 2 separate time points in pregnancy, one of the things we found that was quite compelling to us was a finding where, when looking at asthma as an outcome, we identified theophylline as a metabolite associated quite robustly with it. And subsequently, I'll say we have seen this over and over and over again in all of our cohorts with various study designs. But the reason it hit me was because I knew theophylline as a medication for asthma. and I knew it because this is me in 1984. I was in the hospital taking theophylline and had uncontrolled asthma, and I was hospitalized for quite some time, and theophylline wasn't cutting it. But I also knew that theophylline, while very popular in 1984, was not used today. And so it just begged this question, where is this coming from? And part of my point in saying this is not to live past memory lane. It's to point out sometimes when we're looking at some of these results, connecting potential biology that you know with what you're seeing is important. And so one of the things we quickly identified is that theophylline is a primary derivative of caffeine, and this is a caffeine metabolite. And then I actually remember thinking, oh yeah, that feeling with theophylline is exactly like when I have too much caffeine. But just begging the question, in this case, are there different exogenous exposures that we may want to be thinking of, early in life that are actually, we may only have shown it right now in an association, but could be very easy to implement. If we're looking at the overall disease risk of something, is there something we do that's very easy early in life that could reduce the risk of asthma? And in this case, if you actually look at various studies, even epidemiological studies, the reduction of risk is almost 20% with up to 3 cups of coffee. And this is something I actually think there's a lot more to be learned on from the microbiome. But just simply pointing out this idea that, we can use omics connected with what we understand to understand disease etiology. So birth cohorts is one type of design. Another type of design are disease cohorts that can characterize a disease. And there's many different examples of how this is done. One thing that's been highlighted today, and I think is one of the key ways of doing this, is particularly for heterogeneous diseases, is using the omic data to inform various subtypes. And this is some work done by Rachel and Kevin in this room, where they used metabolomics to identify endotypes. And importantly, again, multiple cohorts, recapitulated those same endotypes in a second cohort, both of about 1,000 people. And what was very striking is they very robustly demonstrated the same clinical differences among these subtypes in the two groups. And so this gets to another point of multiple cohorts and the idea of utilizing cohorts, right? If we can show things robustly, especially things like using omics to create endotypes multiple times with the same clinical outcome, that has meaning to me. Just creating clusters and calling it something, with no level of robustness, to me, I think, it needs a little bit more critical interrogation before we would say that that would be something we would consider actually clinically meaningful. So, when we're looking at disease cohorts, looking at various ways that we can characterize them, I'm just giving one example here of subtypes. There's a lot of things we can do. But I want to spend the next part of this talk going from the idea of, what we see in birth cohorts to disease characterization in disease cohorts, all with using multiple populations, to a study design where we went from really discovery all the way through to clinical translation through a series of cohorts. And this is work that was done in collaboration with Claudia Landenberg, and Priya, who's sitting in this room, is the first author on this paper. And this started with another type of cohort, population-based cohorts. And really the idea was just to look at associations with prevalent asthma in 2 different cohorts, and see what replicated. And when we did this, one thing that was very striking was we saw, among this entire group of steroids, steroid classes, this reduced asthma or reduced odds ratio associated with asthma. And it was very prominent not only in the discovery cohort, but the replication cohort with p-values of 10 to the negative 24. And it struck us, but of course, the very next thing we thought was there's a problem. Okay, so it's good that we have robust results, but there's a problem. And that problem is that for asthma, what do we use to treat asthma? It's oral steroids and inhaled steroids. Now, we had already removed those people with oral steroids, but this idea that what we're seeing is just an indicator that people are taking inhaled steroids was certainly something on our mind. And this point came up again and again. How can we look at this idea of, is this all just confounding by indication, or are there other reasons we're seeing the reduced steroids? So the first thing we did just in our cohort, because we had very good characterization of the medication use, was to look at controls, asthma cases with no steroid use, and asthma cases with steroid use. And if you look up here, you see what we see, which is in every single case, when you look at the group that had steroid use, you saw a dramatic reduction in, you know, the steroid levels that were observed. Again, to David, this is relative quantification. It was a targeted panel, but just keep on thinking relative quantification and we'll move on. So this was something, though, that really made us think. And the reason this made us think, and now again, we're thinking of, at this point, this could be very important. And the reason that this could be very important is because there's a lot of things that may have important ramifications from a medical point of view and even from a developmental point of view. And one of the things was that we observed was, you know, while we see this dramatic reduction of steroids when people are on steroids, even if you were off steroids, it seemed like a fundamental characterization of the disease was that reduced steroids was just sort of a characterization of people who are at risk of asthma. And that makes a lot of sense, right? If you have reduced steroids, your body is not able to respond in the way it would had you had them, especially since it's so critical for asthma. But more importantly is a debate that has been ongoing since the 80s, and that is, while we know that oral steroids cause a reduction of endogenous steroids and can cause adrenal suppression, literature suggesting that inhaled steroids does this has really been controversial, and most people have concluded there really isn't any systemic response. It's just in the lungs. And there's been study after study on this. However, what I'll say is when we look through the literature, these studies are not robust, often done by the pharmaceutical company, and not looking at inhaled steroids over a long period of time. But this question is super important because if people with asthma are on getting adrenal suppression that's significant, this really ends up having a lot of long-term consequences. So we thought to ourselves, well, how can we look at this in a more robust way from a study design? And of course, what we thought of was randomized controlled trials. It's really the only way to disentangle severity of disease and inhaled steroid use. So as this happened, I came up with Lasky-Su's steps towards precision medicine, which you see up here. And we did the first two steps. We identified, we looked in a discovery and validation cohort. We identified a potential cause of the effect. But if we are going to validate that, we really need to look in a more rigorous study design, and that would require a randomized controlled trial. So fortuitous for us, we actually have in our lab probably the best randomized controlled trial of inhaled steroids ever done to date, and I already had the metabolomic data. So this goes to another cohort design. So we looked at population cohorts, and now we're at randomized trials. And so we looked in our randomized controlled trial for inhaled steroids, which was done over 4 years with about 1,000 people. And again, we saw this marked reduction in cortisone and cortisol levels in this trial. Now, we're getting to a point that David just made and I wanted to highlight. Everything done to this point in our study has been done with a targeted platform, but not a quantified platform. And as David said so eloquently, really, in order to determine if this effect is important, we need to have quantification of the result. And that is fundamental, because if this effect is really, really small, it doesn't really matter. So in thinking through this, we questioned how can we actually look and quantify the potential effect of these steroids using clinical standards. And this is a point where I just want to pause and say, we need to be thinking this way, because one thing that we know with all of the data, all of the different types of data we have talked about today, is that we have access to electronic medical records. And when we look at electronic medical records, we have access to a lot of CLIA-approved, fully quantified biomarkers. So can we look at what we've done and maybe our profiles that are targeted or maybe not quantified and look at it in another population where they are fully quantified using clinically approved standards? Because if we do that, we jump forward in where our biomarker discovery is. We are jumping forward many years and many dollars. So we looked in our EMR data and we identified every single person who had a TCH stim test, which is the official test that they use in the clinic for adrenal suppression, and looked at the levels among asthma cases on ICS, off ICS, and various other groups. Now, of course, with cortisol, there's a diurnal variation, and when they do these tests, they are very meticulous about measuring the time of day. And what we saw, I thought, was quite profound. When you have asthma cases on ICS, at every point, they don't even come close to crossing any of the other lines of all of the other people who were compared. And so this to us, in addition to talking to my husband and other of our MD friends about what would be considered a clinically low level, they said this is absolutely low. This is an issue. And so in thinking about this, this then brought us to this next point. Okay, now we're actually demonstrating that ICS in people who've been on it a long time, using all the data we have with an RCT and then capitalizing on the CLIA-approved test for this, really demonstrates that this effect could be clinically meaningful. And so now if you look at Lasky-Su's pathway to precision medicine, we've done all of these steps and we've evaluated the clinical impact and said, yes, this is important. But I want to now think about what these next steps in terms of clinical translation are. And in many ways, this is where we have to rely on others and other expertise, because what does this actually mean? What do we actually recommend to patients? And in talking about this, one thing happened simultaneous to us submitting this paper. And that is, ironically, the Global Initiative for Asthma changed their guidelines for recommended medication use. And they've changed it from being that you should only use bronchodilators for all exacerbations. For the first time in like 50 years, they decided to change their recommendations and say, actually, everyone should just take inhaled steroids. So this change happened within months of us, submitting this paper. And I looked at this and I just thought to myself, like, okay, there's so many things wrong with this from a precision medicine perspective that we don't just have blanket statements, but we'll just ignore that. But they're making this statement saying they're concerned about the effects of bronchodilators without really ever vetting these long-term effects of inhaled steroids. And I want to go back to this picture. And when we think about this idea of clinical translation and the meaning of effects like this, we have to think of it in a bigger framework. I've been on inhaled steroids for 34 years. And from that, the first time you saw this picture until now, inhaled steroids have changed my life. I am sure I have levels of adrenal suppression that are not recoverable. But that's me. I want us now to look at the next generation. This is actually one of my kids. This is him, and steroids have really dramatically changed his life. But what we actually did is we went through and we said, okay, well, we talked to several doctors and then we actually looked at doses of inhaled steroids. And one of the things we found is that when kids get sick or they get a viral infection or they get bronchitis, the doctors immediately give them inhaled steroids and give them high doses, very high doses. And they don't often even take them down to lower doses. And so we asked this question, can we reduce the dose? We aren't saying don't take them, but how efficacious is it to reduce the dose? Because if we can reduce the dose, and that's going to protect people against this long-term adrenal suppression while still being efficacious, that's probably the best way to go. And after talking to several physicians, this is really where we concluded the sweet point is that this really came down to screening regularly and looking at levels in patients to seeing what's happening with their potential for adrenal insufficiency, but giving them a dose, but making it as small as possible with the largest effect. And what we've actually found is that what they've actually said in the literature now is you can reduce the dosage by 50% and still be 90% efficacious. So this to me really was one of these points that highlights this issue, that we really, when we're looking at this and we look at this clinical translation, we may come up with a potential recommendation. We have to be having this engagement with the community and others in the field to actually get to a point where we have that clinical recommendation. So I just want to end with this slide, which is the EMR slide that we started with at the beginning. And in this case, we're going back, but now we're looking at our personal profile. And we've looked at a lot of things in terms of various study designs of asthma, but really putting several together to go from this discovery all the way through a potential clinical translation. And highlight that in this effort today, while the emphasis has been on the amazing power of multi-omic data and the wealth of information, that needs to be very carefully synthesized down into things that we can robustly replicate using methods that enable a level of clinical translation. And we go from complex and we have to synthesize it down into something that we can translate. And that often takes multiple cohorts with multiple study designs that ask incremental questions, getting closer and closer to the disease, and necessarily it takes advantage of multi-expertise among the researchers and collaborators here. But when that happens, I am confident that we will come up with a car that doesn't break down in the middle of the highway. So that I just want to thank everyone here and thank everyone for joining.